DNA alignment is the main operation in computational genetics. If you want to analyze genetic diseases, cancer, evolution, or virus mutations, you should have a general understanding of DNA sequence alignment, because it is a base for any further analysis. DNA alignment plays a key role in getting answers to the following questions. Where inside the DNA is the difference between two species? How to efficiently compare an arbitrary DNA sequence to billions of other sequences in worldwide databases, and which of the various mutations initiates a genetic disease. Instead of going deep into the theory of algorithms, this video gives you a general and visual explanation of key principles, applications, and tools for DNA sequence alignment. To perform an alignment, you need at least two sequences that you will compare to each other. If you have exactly two sequences, this alignment is called pairwise. In the easiest possible case, we have two short similar sequences and we want to compare them. Let's say that one DNA is from a healthy cell and another from a mutated disease cell, and we want to know what kind of mutations happened. Sequences have different length, but they have similar parts here and here. To align these sequences, two gaps are added. Now, blue part of sequences are properly aligned, the letters are on the same positions, but the yellow parts are not. Maybe there's another way to set these gaps. Let's try to move them to different positions. Now, all the similar parts are properly aligned because they are in the same positions, and we conclude that there are 3 mutations, 2 deletions and 1 substitution. Adding these gaps is intuitive for a human, but when a computer aligns millions of nucleotides, it requires quantitative rules. Rules that define which alignment is better. Using the terminology of data science, the distance metric of two sequences. To quantify the distance metric, different algorithms use different metrics. One popular algorithm is Needleman-Wunsch algorithm, and it has the following metric. Each match has a positive score. Each mismatch has a negative score. Each gap has a negative score, and the longer the gap is, the more negative is the score. Numeric values of these scores can vary, and it will affect the performance of the algorithm. For example, the gap may have -2 penalty instead of -1. Implementation, tuning, and improving Needleman-Wunsch algorithm or other alignment algorithms is a broad research topic itself and may deserve an independent video. For application purposes, it is enough to understand that an alignment algorithm adds gaps and tries to maximize the amount of matched letters and minimize the number of gaps. In the previous example, there were 2 sequences, a healthy one and one with the assumed disease. In practice, we would usually have multiple DNA samples, and all of them should be aligned for further research. From a computational point of view, this is a more complicated task, and it has its own name. When there are more than 2 sequences, it's called multiple sequence alignment. The result of such an alignment looks like this. The end result is the same as in pairwise alignment, there are just more sequences. Analysis of the alignment is the same as in the pairwise case. For example, on the second position there are 2 deletions, and on the fourth position there is 1 substitution. Keep in mind that the alignment algorithm affects the result of alignment, thus affecting any further conclusion regarding mutations. One use case of multiple sequence alignment is indeed the identification of mutations and is used in medical research. Another use case is Philogenetics or inheritance analysis, which was used in COVID-19 mapping. It analyzes the overall similarity of sequences. If 2 viruses from different parts of the world are similar enough, it means that they have the same origin and one of them was most probably transferred physically to another location. To define the distance metrics for the similarity between 2 sequences, we can again count matches, mismatches, and gaps. So far we have aligned the whole sequences. This approach is called global alignment and is applicable when sequences are similar. When sequences are very different, this approach doesn't make sense. Instead of aligning the whole sequence, the goal would be to find some similar regions. Look at the following example. There are similar parts, but it is impossible to adequately align them using global alignment. No matter how many gaps will be added, it is not possible to align all the parts, because they are in a different order. It still would make sense to align those similar parts independently. For these cases, there is an approach called local alignment. Instead of trying to align the whole sequence, the algorithm searches for similar subsequences. In this case, local alignment would produce the following result: 3 independent matches. They don't have to be exact matches like in the third alignment. It depends on the parameters of aligning algorithm what is the minimum similarity to be treated as a matching sequence. Quick summary. Global alignment is used to fully align and compare similar sequences. Local alignment looks for similar regions or subsequences and returns these regions aligned. Local alignment is used when there are clearly different sequences and we would like to find out whether they have any similar regions. The most important use case is BLAST, Basic Local Alignment Search Tool. BLAST functions as Google in the world of DNA. It takes an arbitrary DNA sequence as an input, goes through open databases, and finds the most similar matches. It returns the database identifier for found matches and the alignment between the original sequence and the one it found. BLAST is an extremely powerful tool. To ensure adequate time limits, it uses heuristics or smart shortcuts, which means that it is slightly less accurate than other algorithms. BLAST has a user-friendly web interface and also provides APIs. The link for the web interface is in the description. BLAST can generate output search report files in multiple formats, and the most stable is XML. Search report can be downloaded through a web interface or API. If you have a local DNA sequence database, you can connect BLAST to your database and use it as a local search engine for sequences. The general flow of a computational DNA analysis goes as follows. It begins with multiple DNA sequences stored in one or multiple files. We discussed file formats in the previous video, and we can choose, for example, FASTA. The sequences are then loaded into an alignment algorithm. There are various different alignment software, for example ClustalW. The algorithm generates an alignment file which stores the end result of an alignment process. The file format and extensions depend on the alignment software, for example .aln. The file can be stored for further analysis. Alignment software may also generate some conclusions or visualizations. Because alignment as itself is not generally interesting, but rather the conclusions, alignment files are then processed with other software depending on the task. Alignment files also tend to be rather heavy. We will discuss the post-processing and analysis of alignment files in the future videos. You don't have to understand all the possible alignment tools and formats, rather you should know the general framework presented. Alignment files are quite hard to analyze directly, and they are further parsed to other formats for data analysis. But if your research starts from single DNA sequences, you will have to implement the alignment pipeline. You may also be in the situation when you are provided with alignment files which are not yet post-processed. While all the alignment software has different alignment files, one open source Python model, Biopython, provides APIs for manipulating the most popular ones. Biopython can open the DNA files, pass them to alignment algorithms, read the output files, and change the file format of an alignment file. Because it is a Python module, it can be easily integrated as a part of a larger software. Next, I will show you a code for a simple alignment pipeline using Biopython. First, we have to install ClustalW, which is an alignment software. On Linux, it can be done with the following command. Now I import module from Biopython to manipulate ClustalW. In the working directory, I have a FASTA file with multiple sequences. I create a basic command for alignment and then run it. The output is not interesting, but ClustalW has now created an alignment file. To open an alignment file, align IO, alignment input output, is implemented. To open the alignment, I need to specify the file and the file format. Now the alignment is loaded. If I want to, for example, change the file format, it can be done by using align.io, write, and defining output file and file format. For example, here I output the file in Philip format. In the next video, we will discuss how to further process the alignment files for research purposes. References are in the description, and if you have any feedback, please write it in the comment section. See you soon.